difference between biogenesis and spontaneous generation. Now, before there were two theories, biogenesis and spontaneous generation. Biogenesis is the same theory that we believe now, that a living organism can produce a living organism. Whereas spontaneous generation says that a non-living thing can produce a living thing, which is really not possible. Now, biogenesis means making new living things. It is the theory that living things only come from other living things through reproduction. Spontaneous generation means life coming from non-living things. Theory of biogenesis, or evidence against the theory of spontaneous generation. Now, this was the evidence against spontaneous generation. The theory of biogenesis is an evidence against spontaneous generation. The theory says, now the theory of spontaneous generation was disproved by many scientists of 17th, 18th and 19th century. They proved that organisms can be formed from pre-existing ones. This is a note that scientists who experimentally challenged the theory. The name of those scientists were Francisco Redi, Lazzaro Spallanzani and Louis Pasteur. Now coming to Redi's experiment. Francisco Redi, an Italian physician, took the flesh and cooked it so that no organisms were left alive. What he did? He took the flesh and cooked it so that microorganisms can be alive. Then he placed flesh in jars. Here is the jar. What he did? He placed flesh in this jar. Here the first jar was uncovered. The second was covered with parchment and third was covered with muslin. He kept these jars for a few days and observed that maggots developed only in uncovered jar, since it was very easy for the maggots to enter and grow on that maggot, on that flesh. So it only developed on that jar which was open, whereas that closed jar, it was not possible for it to grow, and thus it didn't grow on the closed jars. Now this was Redi's experiment that he did. Spallanzani's experiment. An Italian scientist who disproved the spontaneous generation of microorganisms. Now he was also against that spontaneous generation, which says that a non-living thing can produce a living thing. He experimented that the animals and vegetable broth boiled for several hours. What he did? He experimented on animal and vegetable broth. He sealed it and he boiled it for several hours. And what happened? That it was never infested with microorganisms. From this experiment, they concluded that high temperature has killed all organisms in the broth, and without them, life could not appear when broth were left exposed to air was soon involved by microorganisms. Pasteur's experiment. Louis Pasteur, French scientist, took broth in long-necked flask and then he bent the neck of the flask. He boiled the broth in the flask to kill the microorganism that might be present in them. The curved neck acted as a filter. If the flask with swan neck or curved neck is kept for months together, no life appeared, as the germ-laden dust particles in the air and trapped by curved neck, which serves as filter. If the swan neck was broken off, the broths developed colonies of molds and bacteria. Thus, he showed that source of the microorganism for fermentation, such as milk, sugar and wine, etc., was the air, and the organism did not arise from the nutrients. Now what he did? He took three kind of jars. The first jar, he took broth and it was open, and maggots could easily grow in it. Why? Because it was open. Whereas in the second jar, you can see that it was with a bent neck. Now when in the second flask, it does not grow because the temperature was also high. It also had that curved neck. Now why these maggots are not growing in curved neck? Because the shape, it does not allow the maggots to grow. Even if it tries to go, it's going to trap in the neck, because the neck was really thin for the maggots to enter or the microorganisms to enter. Now in the third flask, what happened? It was again open. What he did, he broke the neck. He broke the neck of the jar, and what he found, that again that maggots could easily grow in it. In the first flask the maggots grow, whereas in the second flask it does not grow, and even if it grows it will be killed due to its high temperature. Even in the jar it does not grow due to its bent or curved neck. When it enters the neck, it prevents the microorganism to enter. The third jar's mouth was open, and thus the microorganism entered the mold. Now all these prove that Louis Pasteur, famous for germ theory of disease and immunology, finally disproved the abiogenesis and proved biogenesis. All these experiments prove the biogenesis against spontaneous generation.